Welcome to this training capsule on animal genetics, which is offered free of charge in collaboration with the LabGenVet website. This capsule consists of extracts of a more complete training session for which you can register at low cost and for which you can obtain a certificate issued by the University of Montreal. The fees collected will be used to finance the hosting and updating of the LabGenVet website so that you can continue using this site as a free and unbiased educational resource on animal genetics. Above all, it is for the genetic improvement of our domestic animals. Happy learning.
Hello, my name is David Silversides. I am a professor and a researcher at the Faculty of Veterinary Medicine of the University of Montreal. I have had a long time interest in dogs and dog genetics, and so that is the topic of today's session. These are the subjects that I'm going to be reviewing. This module is the Genetics of Dogs 1, and we will start with a revision of basic genetic concepts, followed by evolution and the development of breeds, a brief overview of coloration, and I'll explain why it's brief, simple genetic diseases, parentage testing, inbreeding and the coefficient of inbreeding, genetic selection, DNA tests, and DNA profiles. So that's an overview. Now for coloration, I'm just going to give a synopsis of that because I have a second module on the genetics of coloration, the genetics of dogs 2, where I will go into details of the genetics of colors. But that's a topic for another day. In this next section, I will talk about simple genetic diseases in the dog. Once again, I will use the labgenvet.ca website to help us with this discussion, particularly for the dog genetic disease search function and for the section Frequencies of Genetic Disease Mutations by Breed. This is a summary slide that I've already presented, and I will now present it again. 4 simple genetic diseases with a good DNA test, validated for the breed of dog in question. We have the power to eliminate the genetic disease from the breed in 1 generation, eliminate the mutation from the breed in 2 generations. That sounds fast. It is fast. We don't necessarily want to go that fast, but that represents the power that we have this day and age. We have the power, we have the technology. If we lack anything, it's the political will and the education. For the simple genetic diseases of our domestic animals, and that includes dogs, it's fair to say that the majority are autosomal recessive. That means they're not linked to sex. They will show up in both the male and the female animal. And the mutation responsible for the disease is recessive in nature. That means it can be hidden. Once again, we talked about NN, the homozygote normal animal, also known as clear. This dog will not be at risk of the disease and will not transmit the mutation to the next generation. The MN animal is the heterozygous carrier animal. Once again, no risk of disease, but will transmit the mutation to the next generation about 50% of the time. And then we have the animal who is MM, homozygous double mutated, this animal being at risk of having the disease at some point in its life, and who will transmit the mutation to all of its progenitors. Here is an overview of simple genetic diseases in terms of when they present during the life cycle of the animal. And we can divide the life cycle into 3 major parts: the embryo-fetal period, the newborn adolescent period, and the adult period. For the embryonic period, a genetic disease is difficult to identify. It results in ill-defined fertility problems, resorption or spontaneous abortion that could be caused by all sorts of other reasons, not just genetics. So during the embryonic period, a genetic disease is not particularly evident and it is difficult to control. During the second phase of life, newborn to adolescent, a genetic disease is much more evident. For dominant genetic diseases, these are easier to control and very often represent new mutations because a newborn or adolescent animal that is evidently diseased will not be used for reproduction. It is the recessive genetic diseases that are more difficult to control because within the population we have silent carriers, and to have a newborn or adolescent animal affected by a recessive genetic disease, we have to have both parents being silent carriers. The third period in life to mention is the adult animal. Once again, the disease is evident, whether it is a dominant disease or a recessive disease. Clinical signs will often appear after the age of reproduction, such that the bad genetics have already been passed to the next generation. And because of this, mutations are maintained within the population. So in summary, we can talk about simple genetic diseases that are developmental in nature. Form and function do not develop normally, causing the disease. Or we can talk about diseases that occur later on in life. Form and function have been put in place, but are not able to be maintained. And these are degenerative genetic diseases. So, looking at our labgenvet.ca website, the dog genetic disease search function. Click on the link, brings us to this page, which is a database of the simple genetic diseases of the dog where the gene is known, the mutation is known, there is a DNA test available for the disease in question. And this day and age, there are more than 200 genetic diseases of the dog that are characterized with commercial tests available. Our database is dynamic in that you can do searches for a specific disease. You can do searches by specific breed. You can do searches by medical system. Hats off to Dr. Guy Labbé, who, as a fifth-year veterinary student doing an internship at our LabGen Vet Diagnostic Lab, put in place the search functions of this disease database. So now we will look at several examples. I'm going to look at hereditary cataracts in the dog, and I'm going to do my search by disease, and I'll type in cataract. The search function brings up hereditary cataracts and gives me the breeds that are involved. I'm going to circle Australian Shepherd. Now I'm going to do the search by breed. And here's my Australian Shepherd. And here are the genetic diseases that we find in the Australian Shepherd. And here's our hereditary cataracts. And finally, I'm going to do my search by system. And the search is going to be on ocular genetic diseases. Click on ocular disorders, and that gives us the ocular genetic diseases found in the dog. First on the list, hereditary cataracts. Once again, with the presentation of the breeds involved. And here we have our Australian Shepherd. Okay, now I'm going to click on the link for hereditary cataracts. And that brings us to a disease page where we have a description of the disease in question. We have the mutation, we have the transmission genetics, autosomal dominant for the Australian Shepherd, autosomal recessive for the French Bulldog. In addition, we have a presentation of the clinical aspects of the disease. Going back to the dog page on the LabGen Vet website, let's look at frequencies of genetic disease mutations by breed. Click on the link, brings us to this page. First of all, we have an explanation of the analysis that we're going to present with LabGen Vet data, with published data from a DNA profiling lab. LabGen Vet data is generated by DNA PCR amplification followed by sequencing. The commercial lab data was generated by DNA hybridization or profiling techniques. I'll talk about this a little bit later. In the meantime, we're going to analyze the presented data in terms of NN clear animals, MN carrier animals, and MM double mutant animals. And our analysis is for dog breeds. Here we have Australian Shepherd. Click on that, and this is what it gives. the LabGen Vet analysis to 2021. The diseases for the Australian Shepherd. Here's our hereditary cataracts, once again, a dominant genetic disease. For MN carrier animals, out of 91 samples, 25 were carriers, representing 27.5%. For the commercial data, out of 240 tests for Australian Shepherds, carrier animals, 14.6%. So the different percentages just represents the different populations that were tested. Our samples at LabGen Vet represented clinical samples coming from veterinary clinics as well as samples sent in by breeders, whereas the commercial samples represented more samples coming from individual owners. I mentioned Boston Terrier is also affected by hereditary cataracts, this time with recessive genetics. And here are the frequencies we found for the Boston Terrier. Out of 23 samples tested, 4.3% were carriers for our samples. Out of the 69 samples tested by the commercial lab, 7.2% were carriers. Once again, slight difference in the numbers. The point being the mutation is there within the Boston Terrier breed. Okay, what do these mutation frequencies mean in terms of should we perform a DNA test? Yes or no? Here I have a presentation of the risk of having an MN animal. This is a carrier animal, and what that represents in terms of the risk of having an MM double mutant animal. Now, once again, keep in mind that in terms of the disease, if the disease has recessive genetics, this is going to mean one thing. If the disease has dominant genetics, it's going to mean another thing. Let's consider a recessive disease. If we have carrier animals at 25%, in other words, a frequency of 25 carrier animals per 100 animals in the population, that's going to be equivalent of having 2.1% double mutant animals at risk of the disease. If we have carrier animals at 20% frequency within the population, that is equivalent of about 1.3 affected animals within the population. To arrive at a frequency of carrier animals of 18 carriers within 100 animals in the population is equivalent to having one double mutant animal out of 100 animals within the population. Then we can continue to reduce the frequencies of carrier animals within the population. 10 carriers out of 100 animals, about 3 affected animals out of 1,000. 5 carrier animals out of 100, 0.6 affected animals out of 1,000, etc. Going down to 1 carrier animal out of 100, representing just 2.5 affected animals out of 100,000 animals. Evidently, we're talking about a much higher frequency of carrier animals in the population than we are of affected animals. And at some point in time, the frequency of carrier animals is small enough to mean that the frequency of affected animals is marginal. I would say that above 5 carrier animals per 100 justifies doing routine genetic tests for the breed in question. I would say frequencies of carrier animals of under 2 out of 100 for autosomal recessive genetic diseases does not justify performing routine genetic tests for the disease in question. However, if the disease in question is dominant in nature, then that changes the frequencies. The carriers now become susceptible of having the diseases, and frequencies of 1 carrier out of 100 animals, equivalent to 1 animal at risk of the disease per 100 animals, once again justifies routine testing. In summary, for the genetic disease portion of this discussion, there's a lot of information that's available. There's a lot of diseases. Very often they're specific for a particular breed. Use the tools that I've presented. Use the genetic disease search function. To get further information on a particular disease, search the disease by name. To get information on a particular breed, search the breed by name. Use the frequency information to understand the importance of the disease in question. The next topic for discussion is inbreeding. Now, whereas simple genetic diseases represent the genetics that we know the mutation, we know the gene involved, we know the consequences, inbreeding represents the genetics that we don't know. However, we do know the consequences, and we can get a prediction of those consequences by measuring what is called the COI, coefficient of inbreeding. So what is inbreeding? Inbreeding is defined as the breeding of animals that have common ancestors. This will have effects on both the genome and on the phenotype. Inbreeding will reduce the genetic variation of the animal, whereas for the phenotype, it will increase the phenotypic uniformity of the animal. It will increase the prepotency, in other words, the capacity to pass a phenotype on to the next generation, and it will allow a trait to be fixed. However, inbreeding comes with a cost, defined as inbreeding depression, with reduced vigor of the animal, weight, fertility, and vitality, and increased congenital defects, neonatal mortality, and recessive genetic diseases. Once again, our estimation of inbreeding is defined as the COI, the coefficient of inbreeding. On the LabGenVet web page, we have an inbreeding calculator function and instructions on how to use it to calculate the coefficient of inbreeding. Clicking on the link for the inbreeding calculator brings us to this page and the basic setup of the pedigree that's going to be used to perform the calculation, which will be useful for us in performing virtual breedings to determine whether a particular breeding based on pedigrees will increase or decrease the coefficient of inbreeding. In other words, whether a particular breeding is desirable or not. I must mention that the idea and the coding for the inbreeding calculator comes from Benoit Bouchard, a retired engineer and a breeder of pugs. So thank you, Benoit Bouchard. Once again, the calculation is based on this 1/2, 50%. Here are the generations. First generation, the parents. There are 2 parents, and each parent contributes 50% of their genetics to complete the genetics of the offspring. The second generation is represented by 4 grandparents, each one of which provides 25% of the genetics of their grandchild. The third generation is represented by 8 great-grandparents, each one of which provides 12.5% of the total genetics of the offspring in question, followed by the 4th generation, represented by 16 great-great-grandparents, each one of which provides 6.25% of the eventual genetics of the offspring in question, etc. On the LabGenVet website, there are instructions on how to use the inbreeding calculator and how to interpret the results. Once again, the basic mathematics is based on 1/2, the fact that you have 2 parents, the fact that each one contributed half of your genetics. So let's look at a number of crosses that would increase the coefficient of inbreeding. Crossing a parent with a child. In a classical pedigree, it would look like this. Here are 2 parents. Here's the child. The child gets back crossed to one of the parents, producing an individual that will have an increased coefficient of inbreeding. Using the inbreeding calculator provided on the LabGenVet website, a parent by child breeding would look like this. Here's the parent, here's the child, here's the parent being bred to the child, resulting in the offspring that will have an increased coefficient of inbreeding of 25%. Another incestuous breeding would be the breeding of a brother to a sister. In a classical pedigree, it would look like this. Two parents have a male offspring, have a female offspring, brother and sister, who together produce an offspring which will have, once again, an increased inbreeding coefficient of 25%. Using the pedigree function of LabGenVet, this brother and sister breeding would look like this. Two parents produce a male offspring, produce a female offspring. The brother and the sister produce this offspring here, and this offspring here will have an increased inbreeding coefficient of 25%. Finally, we'll look at the situation of a cousin mated to a cousin. A conventional pedigree would look like this. Here are the grandparents, here are the parents, and here are the offspring of the parents, the cousins. Now, if the cousins produce a child, then this child will have a certain amount of inbreeding and a coefficient of inbreeding of 6.25%. Using the inbreeding calculator of LabGenVet, this will look as follows. Here are the grandparents, same grandparents. Here are the cousins. The cousins mate to give the offspring, and the offspring will have increased inbreeding measured by the coefficient of inbreeding. So how to make sense of this? Once again, for our purebred domestic animals, the breeders want a certain amount of uniformity in phenotype, and to get that, they have to have a certain amount of uniformity in genotype. Therefore, a compromise has to be made. However, this compromise is a little bit like the borrowing limits that we have on our credit cards. Yes, we can have a high borrowing limit, and we can have a high rate of borrowing in the short term. However, the debt still has to be paid. Now, cousin-by-cousin matings gives a coefficient of inbreeding of 6.25% with the offspring. We're going to draw a line at about 6% coefficient of inbreeding, and we're going to say that that is equivalent of line breeding, and we're going to say that that is the compromise that we will accept to give us the uniformity of type, the uniformity of phenotype, but still retain sufficient variation in the genotype to avoid the problems of inbreeding depression. Above a coefficient of inbreeding of 6, we can get ourselves into problems with inbreeding depression. And below a coefficient of 6, we will start to have problems with phenotypic uniformity. To go further with this breed-based coefficient of inbreeding analysis, on the LabGenVet website, we provide pedigree-based inbreeding coefficients for dog breeds as calculated by the Kennel Club. Clicking on the link, we have a list of more than 150 dog breeds with their calculated coefficient of inbreeding, once again, based on the Kennel Club's pedigree analysis. In terms of the information that is available to dog owners, dog breeders, veterinarians, with the web, we have a plethora of information. Good information, not so good information, and even bad information. And the trick is to find the good information based on reputable websites. Here I give you a list for dog breeds, for dog coat colors, for dog genetic diseases. Use Google search, use Wikipedia. The kennel clubs are reputable. I've mentioned LabGenVet throughout this presentation. Wikipedia is reputable. For genetic diseases, there are a number of good reputable websites at your disposal. So, thank you for taking this course. Thank you for your attention.